Thanks for the invitation to speak today, Tazib, and welcome everyone. Today's talk is titled Towards a Comprehensive Predictive Model of Brain Health and Dementia Risk. And before starting today's talk, I thought I'd give you a bit of a background as to who I am and what led me to this area of research. So I did my undergraduate degree in neuroscience at UNSW. This is a photo of me from a while ago. But I did that undergraduate degree in neuroscience. I did several volunteer research internships during that time, and I had the intention of pursuing honors, but as part of those research internships, I was working with Neuroscience Research Australia, an institute just down the road from the university I was studying in, and there was an opportunity that arose as a science communicator, and it combined my passions for science and communication, so it seemed like a no-brainer. So I worked there for a short stint of time until I very quickly realized that I was missing teaching. I had a job in my undergraduate degree as a teacher, and I really missed teaching students. I found it really fulfilling vocation, so that led to the Masters of Teaching that I pursued again at UNSW. And soon after, I found that university was the best context for me to be teaching in. So I pursued my PhD here at ANU. So nice to see the familiar faces to be back here again. And so in that PhD, I worked and some of that research I'll be presenting today in addition to stuff that I've done since then. And beyond the PhD, towards the end of the PhD, I got a job at the University of Canberra as a lecturer, and I've worked there ever since. And really, these photos are here to give you timestamps of not only my background, but I think it's very apparent as to why I'm an aging researcher. I've been ravaged by the time of age, and so there's a lot of personal interest and motivation, understanding the process that contributes to aging, and hopefully I can slow it down in myself and maybe in others. And so broadly, I mean, Tazib did a lovely introduction, but broadly, my background is I'm a lecturer at the University of Canberra, and my role consists of both teaching and research. So it's sort of the dream job for me. And in addition to this, I lead a community of practice at the University of Canberra, which focuses on generative AI, and it helps upskill staff and students to understand how to use the tools effectively in their research and learning and professional practice. I've developed a series of workshops that are available on this website of mine that you can access through the teaching tab. So if you can spell my name, anandinambikaraj.com, you can access these materials. But also Tazib will circulate these slides as well. So I'm sure that'll be available. And so broadly, that's who I am and things about me. And for today's talk, I wanted to really focus on the questions that motivate me in research, that inspire me to do the research that I do, and what I commit my time and efforts to for the projects that I work on. And so fundamentally, this starts with understanding what are the causes of dementia. And this leads to an investigation of the mechanisms that contribute to aging and the pathology of dementia. Depending on there are different ideologies within this space in regards to aging and dementia. Some people think that it sits on a continuum. Others think that there's some degree of overlap, and others believe that they're completely separate processes that are happening. And I think in some part, these discussions are in some ways confounded by lifespan. When I ask myself the question, if everyone was to live to 300 or 500 years of age, would they develop dementia? From what I understand of the literature, my thinking is that the answer would be yes, if not at that age, much earlier. And so, of course, theory of evolution would posit that there'd be some sample within that population that would be resistant. But broadly speaking, across the whole, we would see pathology similar to dementia developing. And so this speaks to an investigation of the mechanisms that contribute to aging and understanding dementia, but also it informs a lot of how I think about this research space in terms of the life course model that I adopt when I think about research questions. So in addition to this, much of my work focuses on genetics and environmental and lifestyle factors, and there's a particular focus on cardiometabolic factors. I think the saying that what is good for your heart is good for your brain rings true, and we see this in terms of Dementia is a broad umbrella term that consists of many different subtypes, Alzheimer's disease being the most well-known, but followed closely by vascular dementia. And the overlap between Alzheimer's and vascular dementia in terms of the risk factors rings true to the importance of the magnitude of effect that cardiometabolic factors have to brain health. And then this follows as well in terms of thinking in terms of the largest factors which have the largest effect. Sex-specific factors emerge, and I'll be discussing these things in more detail throughout the talk. So that's probably the theory that drives much of the research that I do. And the next focus question that builds from that theory and closely tied to it is how can we effectively utilize available resources to reduce dementia risk? And so this starts with the development of accessible measures of brain health that accurately predict dementia risk. If you wanted to have an assessment of your brain health right now today, you may give me your own subjective opinions about how your brain is performing, but typically the best measures that we have available are not accessible to everyone. These include neurocognitive assessment and neuroimaging. And so there's a lack of an ability for the general public to access measures, to understand how their brain is performing throughout the life course, and perhaps identify stages at which there's decline occurring. And so there's several measures out there that are possible candidates, but I won't go into too much detail right now, but just the focus on much of my research is trying to identify what accessible measures could be. And so this leads into the development of predictive models across the life course that quantify dementia risk, which are meaningful at an individual level. I think that life course approach, you already know from what I've said thus far as to why that's a focus. But when it comes to many of the prediction models that are available, I think when I look at the literature, there are many. The first question I ask myself is, what does this model tell us beyond what I already know as someone immersed in this field that I could tell to the general person off the street in terms of eat a nutrient-rich, varied diet, engage in physical activity, have a rich social network, all of these factors which we know are good for the brain. You try to stop smoking if you smoke, or eliminate smoking, or reduce alcohol intake. What is it that these predictive models provide to us over and above these generalist statements that we know from population level data? And I think this is a major gap in the field in terms of individualized level of prediction for brain health and dementia risk. And then from this follows to explore targeted interventions that improve brain health or minimize the rate of decline so that we could delay the onset or progression of dementia. So that's really the core focus of the translation of theory into practice. And a third facet of the questions that motivate what I do is how can we effectively engage the public in scientific research so that they can make informed decisions about their health? As you already know, I'm a passionate science communicator, and I think in part it's because of when we have an informed society, an informed public, I think it leads to one in which we make better decisions as a whole, and people can learn more about things they may not already have access to. And so this includes working with policymakers, health professionals in the community, and those with lived experience. And admittedly, I don't do all of these things well. I'm still growing my connections and networks with policymakers and health professionals. And I'm part of the Center for Aging Research and Translation at the University of Canberra. And there's several researchers there who do a really inspiring work in this space, in addition to working with people with lived experience of dementia. But what I think I do excel in is in the teaching in science communication. I view teaching as an avenue to provide impact in the minds of students that allows them to think critically and scientifically about what they see, not only in science, but the world around them, and that has a direct impact in society. And science communication, whenever I engage in whatever research I'm doing, the pipeline doesn't end with publication. I try to translate those findings to the public, and you'll see elements of that in my talk today. And so broadly, those are the questions, and these are the reason why I start this talk with this slide and this level of depth in these questions is because you'll see that much of the research I present today, the common thread that ties it all are driven by these questions. And so hopefully you can keep that in your mind. But as a background for dementia, it's a growing issue, and the these are estimates that show the incidence by 2050. Global incidence is estimated to be over 130 million for people living with dementia worldwide. But what this picture doesn't really tell is how these estimates differ by sex. And so when we pull data from one of these countries, say Australia, living with dementia, the estimates between 2023 and 2058, what we see is that, of course, in men and women, in the lower two blue lines, we see that there's an increase in the incidence of dementia predicted. But we see that women are disproportionately affected by dementia than men. And this increase occurs across the lifespan. And when we look at the underlying causes of death in Australia by sex, the picture again becomes apparent that women, on the right of this slide, we see that the number one cause of death in women is dementia, including Alzheimer's disease, followed by coronary heart disease. Whereas in men, it's the opposite way. Coronary heart disease is the number one killer, followed by dementia. And so for a long time in the literature, it was thought that this is largely attributable to lifespan. One of the biggest, or the biggest, risk factor for brain health and dementia is lifespan. The older you are, the more likely it is that you'll develop dementia. When we look at life expectancy for Australian women, this is 85.3 years, and men it's 81.2 years. So we have a difference of about 4 years. And much of the differences we see in the literature were attributed to this difference in lifespan. And this is consistent with global trends, which show that on average, we see that women live longer than men by approximately 5 years across the world. However, there's some interesting research, including this paper conducted by Nichols and all others, that showed that age-standardized prevalence, global prevalence of dementia in females was 1.17 times the age-standardized prevalence in males in 2016. And what this says is that age is a really important factor in the story in terms of the difference that we see between men and women, but it doesn't explain the whole story. And so much of my early research was trying to investigate, well, what are the factors that could contribute to this sex difference? And there's other evidence as well that seems to indicate sex-specific effects when we look at brain health and dementia risk. So, for example, apolipoprotein E is a gene that we all have, but there are different alleles, E2, E3, E4, with the presence of the E4 allele, a risk factor for dementia. And whilst the relative frequency of this doesn't differ by sex, there's a meta-analysis that indicated that women with ApoE3 and E4 genotype, they had an increased risk of Alzheimer's disease compared with men between the ages of 65 and 75. So we see these sex-specific effects emerging in perhaps sensitive periods across the life course. And in a longitudinal study of the ADNI study, the Alzheimer's Disease Neuroimaging Initiative, it found that for those with mild cognitive impairment, the precursor stage, it can be thought of to Alzheimer's disease, although not all convert. Cognitive decline was faster in women than men. And these models were adjusted for what you'd expect them to be adjusted for when looking at brain health as an outcome. So age, ApoE status, education, and baseline cognition as measured by the MMSE. And so already we're seeing sex-specific trends occurring between men and women in terms of either Alzheimer's disease risk or cognitive decline. And the question then becomes, where in the life course do these trends begin and accentuate through sensitive periods? And this is a beautiful study conducted in 2022, which charts it provides a brain chart for human lifespan. And these charts, if you've had a child, you'll know that much of their developmental milestones are mapped to normative data and charts. So whether their height, their weight, how they map to normative data. And in the same with neurocognitive assessment, this is also mapped to normative data. And what we're missing in neuroimaging research prior to this paper was a similar sort of conceptual idea for brain health. And so this study aggregated over 120,000 MRI scans from across more than 100 primary studies, had over 100,000 participants from 115 days post-conception to 100 years of age. And you can see included in this range are things like the UK Biobank study, which you'll hear more about today, which has participants from 45 to 70 years of age at baseline. And what they found, which was really interesting, is in this slide, you'll see what I want you to focus your attention on is, in particular, the gray matter volume and subcortical volumes. We can see that on the x-axis, we see age from birth to 100 years. And what we see is a decline in brain health in late adulthood and middle adulthood. But what's really, really interesting is, particularly for gray matter volume, we see that this extends even earlier in young adulthood and perhaps even adolescence. So this speaks again to evidence of perhaps a life course approach where the changes that are occurring earlier in life are contributing to our brain health in later life. And so this is also represented in different ways. And in this slide, what I want to draw your attention to is the intra and inter variability that we see between sex. So men are represented in teal dots and women are represented in the red dots. And what we see is that there's variation between sexes. For example, in that first row, you can see in say, gray matter tissue, we can see there's variation between them. But also within sex, we see variation as well. And so this speaks to perhaps different exposures to risk factors, different genetic predispositions, but also something to keep in mind is the noise of data that we work with when we conduct models. And predominantly when we do regression modeling, we're dealing with average estimates of a group. And so we have to always consider how noisy is the data around that average. And so that's something that is a theme I want you to keep in your mind, not only for this talk, but also when thinking about research in this space and others. And so the ideology of sensitive periods throughout the lifespan in terms of those brain charts, what's interesting, a paper published in the UK Biobank found that there was a slight acceleration of hippocampal volume loss around the ages of 60 to 65 for women. And so that's elicited in slide A for hippocampal volume. You can see that men are in the gray bar, and in the orange bar, you see women. And you can see that the rate of decline in hippocampal volume is quite steady for men. But in women, there seems to be, it's a steady state, and then there seems to be quite a rapid acceleration around 60 to 65. And also, if you look at slide C on the bottom, we can see that for both women and men, there's an increase in rate of hippocampal volume loss relative to the rest of gray matter from around ages 67 for women and 63 in men. And so the authors of this study posited that, well, maybe the hippocampus has particular vulnerabilities to aging, as these effects were not detected in neighboring brain areas, including parahippocampal gyrus and temporal gyrus. And in dementia research, this has particular relevance given the fact that the hippocampus is one of the first regions that's impacted by Alzheimer's disease. So hippocampus is important for memory formation and spatial navigation. It's the site that's first impacted and where it can be a useful predictive tool of risk for Alzheimer's disease. Now, the evidence is not as clear as I'm articulating when we look at other biomarkers. So for example, when we look at amyloid beta and tau burden, I could certainly present papers that show a sex difference, but on aggregate, when we look at the available evidence, I think that the evidence regarding sex differences is limited, and it really requires further replication. And in part, the reason why the evidence is limited is because often in neuroimaging studies, analyses adjust for sex, so they include sex as a covariate in models, but they don't often include sensitivity analyses that stratify analyses by sex or investigate potential sex interactions. And as an example of this, there's a meta-analysis that was conducted in regards to sex differences in contribution to brain reserve. Now, brain reserve in this study was defined as consisting of IQ, education, occupation, cognitive activity, multilingualism, socioeconomic status, physical activity, social support, or marital status. Across all of those measures, they identified 16 studies that included an analysis of sex. So it speaks to the fact that more work is needed in the space of differentiating effects as a function of sex, particularly when you think about the intergroup variability that we see highlighted previously. So what are the reasons for the sex differences that we've discussed thus far? Well, perhaps historical inequities, which result in disproportionate access to education and occupational opportunities contributing to brain reserve. There's also the potential selective survival bias in men, with men who live beyond 65 years having a healthier cardiovascular profile. If you recall, the number one leading cause of death in men in Australia was cardiovascular related incidences. And so therefore, if they're less likely to develop Alzheimer's disease in later life because they have a healthier cardiovascular profile and they've lived through midlife. There are possible interactive effects between sex or sex-specific factors and genes, which we'll explore a bit today in my research, and also unique neuroendocrinological processes in women, including menarche, menstruation, pregnancy, and menopause. And so much of my early research started by focusing on menopause as a candidate to explore in terms of the contribution to these sex differences that we see in brain health. And so menopause is a brief overview. It comes from the Greek words meno, which means month, and pause, which means stop, thus indicating the end of monthly cycles or menstruation. And it's historically been understudied in the context of aging. So over a period of 23 years, peer-reviewed neuroimaging articles which focus on menopause accounted for approximately 2% of the aging literature. So something that affects approximately 50% of the population, and in the research space is accounted for 2% of it. Now, the average age of menopause lies somewhere between 46 to 52 years of age. And so given that life expectancy of women in most developed countries lies around 81 years, this gives you estimates that approximately 40% of women's lives are in a postmenopausal state. So more research is needed, and it's given its contributions to the brain haven't been well explored, it seemed like a reasonable place to start. And it was not as easy as just starting. What I realized from cursory scan of the literature is that the definitions used for women's health are not consistent, and perhaps relate to the fact that it's not widely studied or studied in relative sense to other areas. And so when it comes to the terminology associated with menopause, so menopause, it's defined as the absence of menstruation for 12 months. And so it's only defined in retrospect. And so what you see on the right is an adaptation of a figure from the Stages of Reproductive Aging workshop, which aimed to sort of try to develop a standardized way of defining the stages for reproductive aging, including menopause and premenopause. And what I did was I tried to see, well, how well were these criteria, which are deemed as the gold standard, adhered to? And we found that there's, unsurprisingly, to some extent, quite significant variability, but the degree of variability was what was surprising. So there were major inconsistencies in premenopause as a definition, and in comparative sense to postmenopause. And to some degree, I think this is in part evident by this slide when you see that the term premenopause isn't really operationalized in this stages. And so it led to different researchers having their own definitions. Why is this so important? Well, because if there's not standardization in terms of definitions, then it's difficult to compare results across studies, and it has flow-on effects when trying to understand the effects that we see in different stages of women's health. And so this review not only investigated its uptake and found these results, but also proposed suggestions for ways to clarify it in addition to operationalizing premenopause. And so once I'd navigated that space, I was interested in, well, what are the relationships between menopause and the brain? So that first led to an investigation of, well, what changes happen around menopause? What are the biological and physiological changes around menopause? And the focus here for me were cardiometabolic factors, largely because of its influence on brain health and its magnitude of effect, as we discussed earlier. And so I focused on fat mass and lipids initially. Now, in this space, there's a really difficult question to tease apart, which is, how do we know that any effects that we see are attributable to menopause versus aging? Because as people age, they co-occur with menopause, particularly in midlife. And so they're very closely correlated. So how do you tease apart effects attributable to aging versus menopause? And so much of my research in this space used careful statistical designs to try to give us a sense of whether effects were attributable to menopause or aging. And so the first study, which I identified in this space, was to look at fat mass changes around menopause. I mean, fat mass is critical for brain health, and it's a central component of metabolic disease. And what we found, well, what we looked at initially were a whole bunch of measures of fat mass, because in addition to menopause nomenclature having varied definitions and the challenges with delineating between aging and menopause, which was a challenge in this space till the systematic review, the addition was the use of fat mass measures. BMI is a very useful measure for quantifying changes in the body mass, and its self-report measure of BMI closely links with the measured BMI. But what we find is that its measure of fat mass is not as clear, and it's not the gold standard comparatively to other tools such as DEXA, which is the gold standard of measuring and quantifying different compositions of body fat, bone mineral density, and lean muscle mass. And so this study looked at a whole bunch of fat mass measures. You name the fat mass measure is likely studied here: waist circumference, waist to hip ratio, BMI, fat mass, trunk fat as measured by DEXA. And so it resulted in 200 studies, cross-sectional studies, and several longitudinal studies being included, about 11 longitudinal studies that were included. And the sample size for the cross-sectional analysis was over 1 million women, and for longitudinal about 2,000. And what we found is that in this forest plot here, we found that postmenopausal women had larger trunk fat than premenopausal women. And this equated to, in this forest plot, about 5.5%, which was estimated to about 0.5% per year of a difference. And so the total fat mass significantly increased between pre and postmenopausal women. But when we tried to tackle how much of this was attributable to aging by including aging in meta-regression models, we found aging actually accounted for a large proportion of the unexplained variance in fat mass. And as a brief aside, I just want to point out that since this paper was published, there's been a really interesting paper that I've cited here that has found that typically when people talk about metabolism, and what is aging and the changes associated with aging in terms of capturing this variance, it's sort of, it would be its own talk in itself. But what I want to point out is that often people associate with aging a slow rate of decline in metabolism. But what this paper found was that, in fact, metabolism remains stable in adulthood between 20 to 60, and it declines beyond that point at 0.7% per year. So it speaks to the fact that the changes that we see, or the increases in fat mass as a result of aging, are maybe attributable or largely attributable to a decrease in physical activity, increase in sedentary behavior. People have jobs where they perhaps sit down 9 to 5, so it takes out times in their life. So all of these factors and stressors of life that increase with age contribute as well. So when I think of aging, I don't want you to think of it as a non-modifiable factor in this context. There's modifiable factors that could contribute to it. So aging accounted for unexplained variance in fat mass, and the other strategy we had to just see if we could detect an effect attributable to menopause, we looked at longitudinal trajectories for changes in women, and in studies that included women from 18 to 45 years of age, and compared these with our trajectories that we found in our systematic review around menopause. And we found that the trajectories across the lifespan didn't really differ when we focused on menopause versus the longitudinal studies, which seemed to indicate to us that for total fat mass, there was not really a detectable effect of menopause. And again, we conducted an interaction to look at age and menopausal status, and we detected no interaction effect there. But what was interesting as well was when we looked at the distribution of fat mass, we did find a change in fat mass distribution with increases in central fat and decreases in leg fat. And we posited that this might be attributable to hormonal shifts around menopause. So higher testosterone to estrogen ratio, so it may have contributed to enhanced central fat deposition. And so to test this, we did sensitivity subgroup analyses based on hormone replacement therapy use. And so we looked at women who were using hormone replacement therapy, and there was a significant increase in body fat, total body fat, and a significant decrease in trunk fat in women who used hormone replacement therapy, which suggested a possible protective role of hormone replacement therapy in preventing or reducing trunk fat deposition, although not in preventing overall fat mass gain. And there are many factors that we posited may contribute to this. So it may be that women who use hormone replacement therapy are healthier. Typically, they're of higher socioeconomic status, higher education. These factors can contribute to fat mass. But what was interesting is these findings are consistent with the previous meta-analyses of 8 randomized controlled trials, which found postmenopausal women using hormone replacement therapy had less waist circumference and trunk fat percentage compared to placebo. So it seems consistent with the indication that the change in hormonal profile around menopause may contribute to what we see in the distribution of fat mass. And so the second cardiometabolic factor that was of key interest for us was of lipids. And we tried to precisely quantify the changes that occur around menopause in lipid levels. A challenge in this area was the fact that we didn't have sufficient longitudinal studies in this analyses, but we used different techniques to try to quantify the differences that we could see. And so in the cross-sectional studies, we found that age explained some, but not all the differences in lipid levels between pre and postmenopausal women. And so we did a sensitivity analysis of those with a mean age difference between pre and post of 5 years or less. And we tried to see were there differences detected there. And there were no significant differences in magnitude, direction, or significance of effects compared to initial estimates for different measures of lipids. And what I want to draw your attention to in this slide is that the difference in high density lipoproteins, there was no difference, as you can see quite clearly in this forest plot. And so it may suggest that for these other measures of cholesterol, there's an effect of menopause, but it could also be other factors, including group differences that you see, or given insufficient longitudinal studies were available for meta-analyses. And so given this, the focus now that we've precisely quantified the cardiometabolic changes that we see around menopause, the interest was now to see, well, what is the association between these cardiometabolic factors and brain health? And so in this study, we focused first on fat mass. And a key design feature of this study was we wanted to quantify changes in fat mass relative to baseline fat mass. And what I mean by that is an assumption we had is that an increase in one BMI point at 20 may not be exactly the same as an increase in one BMI point at 25 versus 30 versus 35. And so we wanted to embed within the design a way where we were capturing the changes from baseline. And so we used longitudinal data, and we used the UK Biobank study, which is a large study consisting of over 500,000 participants, of which at this time had approximately 20,000 in terms of neuroimaging scans. And we looked at these changes in terms of standardized thresholds for categories of whether people were normal at baseline or overweight or obese stable at baseline. And so we saw whether they were normal throughout the follow-up in terms of their weight range, whether they were normal initially at baseline and then proceeded to gain weight to obese, overweight, or whether they were overweight, obese, and lost weight over the follow-up, or whether they maintained the weight range at around overweight and obese by these thresholds. And what we found was that individuals with chronic overweight or obesity, defined by maintaining that level across the follow-up period, they had significantly lower hippocampal volumes when compared with those who maintained a normal level of fat mass at baseline and follow-up. And so the average follow-up was about 7 years here. And individuals who are within normal range of fat mass at follow-up assessment, yet were previously classified as having overweight or obesity at baseline, had lower hippocampal volume. So that was really quite a surprising effect that we saw there, that even though at follow-up they presented within these ranges of what was deemed as normal, it was still observed that the prior history of fat mass seemed to be associated with the changes that we see in the brain. And so this led us to conclude that perhaps the detrimental effects of overweight and obesity extend beyond the duration of overweight obesity itself, which emphasizes the importance of maintaining a normal weight for brain health. And so I want to put these estimates in the context of what they should mean. I think we should always focus on magnitudes of effects and what they mean in terms of not just saying significance or higher or lower, but what do the size of these effects mean? And so what we see here, that for the differences between groups of about 1% for those with overweight, obese, stable to normal weight. They equate to, this is a plot I drew up from data from these two systematic reviews, which quantifies the annual change in hippocampal atrophy below 55 to 70, 70 plus, and then those with Alzheimer's disease. And what we see here is that the estimates found in this study equate to approximately 1 to 3 years difference. And I give you that range because the participant range goes from 45 to 70 at baseline. So it gives you a sense of what that difference equates to. And so what was interesting as well beyond this is we looked at multiple measures of fat mass. So waist circumference, waist to hip ratio, and also total body fat, sorry, BMI. And what we found is that the largest magnitude of effect was consistently observed for waist circumference. And we posited this is likely because of its correlation with visceral fat, but we wanted to conduct further analyses to sort of test this hypothesis. So we also found that in subgroup analyses in women, they consistently reveal lower hippocampal volumes for those who were overweight, obese, stable across the follow-up period, overweight, obese at baseline to normal, and normal to overweight, obese at follow-up, compared to those who are normal, stable for waist circumference. However, the results were not consistent across waist to hip ratio and BMI. And to us, it spoke to the utility of waist circumference as a measure of visceral fat, and it may reflect the changes that we already discussed in terms of what's happening after menopause with the accumulation of central fat. And so what we see here in this slide from the study is that BMI for total body fat as measured by DEXA, BMI is actually a really good index of total body fat with a correlation of close to 0.9, followed by waist circumference. But when measuring visceral fat, we see that waist circumference outperforms BMI with a correlation of 0.8 comparatively to 0.6. And I think this speaks to the effects that we see, how they're consistent in waist circumference and the mechanism of action between changes in fat mass and the brain with pro-inflammatory cytokines. And so in terms of cardiometabolic factors in brain health, I've walked you through in extensive detail fat mass as it's a central component of metabolic syndrome. But in addition to that, colleagues and I looked at waist to hip ratio, high density lipoprotein, low density lipoprotein, and blood pressure. And we found a consistent story emerge when we compared pre and postmenopausal women in the UK Biobank on these cardiometabolic factors. The brain health outcome in this context is white matter hyperintensities. And it was what we found is that postmenopausal women had poor cardiometabolic profile as reflected by these measures compared to premenopausal women beyond the effects of age. And this was indicated at the poor cardiometabolic profile as higher baseline levels of blood lipids, blood pressure, long-term blood glucose, as well as longitudinal changes in body mass index and waist to hip ratio. So all of these were associated with larger white matter hyperintensities. And so after investigating all of this, a key question was, well, how do we quantify the changes that are occurring around menopause and its relationship to brain health? So using the UK Biobank, I was interested in several reproductive factors in women, but for the purpose of this talk, I'll focus solely on the findings for menopause. And what was of interest in this study, where we were looking at differences in brain health and trying to grapple with the statistical challenge of whether those differences were attributable to aging versus menopause, what we found is that postmenopausal women experienced 0.23% greater reduction in total brain volume than premenopausal women for every 1 year increase in age. And you can see that in this plot on the far left, that the slope, postmenopausal women are in teal dots and premenopausal women are in red. And you can see the slope is much greater on the left, which represents total brain volume. However, this interactive effect was not detected for the hippocampus. For postmenopausal women, what was surprising and contrary to our hypothesis is that every year delay in age of menopause was associated with a 0.32% smaller total brain volume and 0.31% smaller hippocampal volume. And what was even more surprising was the fact that when we look at these lines and these average estimates, that postmenopausal women had 1% larger total brain volume and 2% larger hippocampal volume than premenopausal women. And this effect was consistent across all analyses. And it really was quite confusing. And so we analyzed it in different ways. of course, multiple regression models, which controlled for age. We conducted propensity matching, which used exact matching for age, and also the age-restricted analyses between 45 to 55. And we found consistent results across all in terms of postmenopausal women having larger brain volumes than premenopausal women. And so what does this speak to? I think it speaks to a few factors. One is the fact that first, this is cross-sectional data, and so we're looking at differences across time. Second is the nuances of the data set, as with every data set, but with the UK Biobank, there are particular nuances in the sampling that may not be representative of the larger population. And finally, I think what I want to draw your attention back to is the point I made earlier about the noise of data and the between and within group variability. These are average estimates quantified across very noisy data. And so we need to find ways to sort of capture this noise better to allow us to make informed estimates and understandings. And so that's sort of what I've attempted to do in further research, and also try to tease apart the effects that we see attributable to possible confounders. And these include women who have used hormone replacement therapy, who were excluded in the previous study because of its confound in terms of self-reported age of menopause. And so in this study, we looked at the interaction between genetics, age, ApoE status, and brain health. And what I want to draw your attention to is just the results that focus on ApoE and hormone replacement therapy. In simple terms, does the relationship between hormone replacement therapy use and brain health depend on ApoE status? Keeping in mind that the E4E4 and E4E3 variant are those that are predictive of Alzheimer's disease. And what we see in the cognitive measures in the top here in these box plots is that, in fact, there doesn't seem to be any difference that can be seen across groups for these different ApoE variants as a function of hormone replacement therapy use. This study in alone wasn't surprising, but given the context of the field, it was very surprising, because there are several papers which seem to indicate the opposite of what this study found. So here's a paper by Yaf, where the abstract notes that there's an interaction between estrogen use and E4 presence with a p-value of 0.037. A study by Sala and colleagues which showed that ApoE users, there was an interaction that was present with a p-value of 0.009. And also Kang seems to indicate that they detected an interactive effect as well. However, when you read these papers in further detail, a clearer story seems to emerge. So for example, in Yaf, in their analyses, after they adjusted for key factors such as age, education, race, and stroke history, the interaction between ApoE and estrogen use was no longer detected. However, they stated it lessened somewhat in statistical significance and only report the outcome from the first unadjusted model in the abstract. In Sala's paper, when you go to the table that shows the findings for the interactive effect, notice that there's a little letter A next to the 0.009, and the text below the table states insignificant after correction for multiple comparisons. So given the number of analyses, the effect was no longer significant after this fact, and it was for a specific domain within the cognitive test. And this isn't anywhere to be noted in the figure that you see on the right that depicts an interaction term or in the legend below it or the abstract. And in Kang's paper, when you look more closely, the interactive effect is actually described as a suggestive interaction. It doesn't meet the threshold for statistical significance. So why am I raising this point? Well, this isn't an issue with these papers, but it's actually more broadly, this is ubiquitous in science. This reporting of results really matters, not only by the scientists, but also by journalists. And what we see here are two news articles that report hormone replacement therapy could cut Alzheimer's risk in some women early study, right? Or could ward off Alzheimer's amongst at-risk women. And both of these studies are citing the Sala paper, which was the 0.09. It wasn't significant after multiple comparisons and was only a small domain of one cognitive test. So nothing really directly related to Alzheimer's disease. But these narratives are articulated. And I think this is why it's really, really important to communicate science and scientific thinking to the public. And so that's what I attempt to do with my research. I engage in news and media through the university's media teams, and the work that I do as well with CARET, Center for Aging Research and Translation, where we try to communicate this to the public. Now, it's difficult because in science is challenging, and you're trying to distill the essence of those findings to the public. And also there's layers of media that interact, that try to change the messaging some ways in different forms. And so this is why I engage in all forms of media, but in particular, I enjoy podcasts, and I have my own podcast called Midnight Conversations, which aims to pick a paper and discuss those findings to the public. And I find it's a very useful way to sort of make science accessible to the general public. It has over 55 episodes, 7,000 plays, and is available on Spotify and Apple Podcasts. And so there are ways in which we can have impact as researchers beyond this, and I think science communication matters for this fact. And when I tell people what I do, number one question I get asked is, what can I do for my brain health? What are the things that can help ensure that I don't experience dementia or I delay it beyond my lifespan? And this paper, or these series of papers from Livingston over the years, is a good start of trying to quantify the estimates, because I think we should be quantifying the magnitude of effects that we see and directing behavior in those that are will make the largest impact. And so what we see here, what I want to draw your attention to, in addition to the percentages that we see for the magnitudes of effects in terms of if that factor was eliminated, the percentage of reduction in dementia prevalence that we see, is that over the years, from the left side, 2017, when the first paper came out, to very recently, 2024, is the shift that we see in risk factors that are from initially a large focus on late life to midlife. And if I was to guess where that would go, is that it'll shift earlier and earlier, because there's already very good research showing the relationship between fat mass in early life is predictive of the effects that we see in midlife and attributable there. And so it gives, but it's a good starting point in quantifying these estimates. But always we should be skeptical of estimates that don't have levels of precision around them, right? So estimates should always have that level of uncertainty, given the noise and data that we know is always there. And so in addition to the shift from late life to early life, other predictions I have are the inclusion of emerging factors. Some research done by my PhD student here, Turgal Namsrai, looks at sleep and brain health. And the paper on the left is a paper which looks at the UK Biobank data set, and on the right is a recently published systematic review, both showing consistent findings in regards to the contribution of sleep to brain health, with examples of poor sleep defined by or sleep less than 6 hours or greater than 9 hours, resulting in impaired brain health on metrics like structural volumes. And so we see that brain, and mechanistically, this makes a lot of sense. Sleep is really a critical time in which your body is clearing waste products from that's occurring in the brain and the body. And so sleep, I think, will be an emerging factor of interest in this research space. So really great research being done here. And there's much more work to do when we're thinking of what are the emerging risk factors for predicting dementia risk. And so here's a study that uses the UK Biobank and tries to develop an index to predict dementia risk. And what I want to show here is the fact that it's called the UKB DRS, and it compares its performance to other well-known dementia risk indices, including K, DRS, and ANU-ADRI. And what we see in this plot on the right is that the line in green is the predictive power of age alone in the model, just age included in the model. Whereas here, the other lines show other indices that include different variables and factors. And what we see is that age does a really good job of predicting dementia risk over and above some indices. And the black line that you see here is age, few other factors, but also the inclusion of ApoE. So age and ApoE are really the largest magnitude of effect. And so what do we need to do in this space to improve prediction? So one is a focus on emerging and existing risk factors and quantifying their estimates, but also a real limitation in this area is that many of these models are developed with a frequentist framework. And we know already from even this study here in Livingston that there's well-established risk factors for brain health. And so we can incorporate that prior knowledge through Bayesian analyses to allow for what I think would be more sensitive models that could be updated as emerging evidence comes in. And so I think that's a really critical area, and that's one that I'm working on in this field, is in developing these models and improving their performance. And what I'd like to do, like in addition to the research questions I've focused on there, one focus was on a accessible measure of brain health. And for me, what's accessible is one in which people could walk into a GP and get an assessment as to how their brain is performing. And so we already know that there are risk factors that are well correlated with brain health, including fat mass, blood pressure, family history of dementia. And so but an unresolved question is, what is the time at which people interact with the GP when they experience symptoms? And so I'm conducting research with collaborators at University of Sydney and at the Brain and Mind Institute there with Frontier. And this is research that I'm currently conducting, and it's soon to be submitted for publication, that looks at the time it takes for people to see to obtain a GP referral from first self-reported symptom onset. And what we find is, and this data set is a longitudinal data set across 16 years of those with Alzheimer's disease, but also other, the clinic is focused on frontotemporal dementia. So these acronyms you see there relate to those variants. And what we see is that for those with Alzheimer's disease, it's typically about 3 years. And this uses Bayesian models, which was really useful in terms of using the prior knowledge in this space to inform these estimates. And what we find is that this estimate here is on the higher scale than previous estimates. And it actually becomes quite clear when you look at these density plots across the different diagnostic groups. And we see that for most people across most diagnoses, people see a GP within the first few years, but there are groups of people who tend to delay that visit to the GP. So those trends are really interesting. And with Bayesian models, you can specify the distribution, which allows us to model these things really accurately. And so what we see as well in the plot on the left is that with relative to Alzheimer's disease, which is centered on one, we see that there's relatively not a lot of difference between many of the variants. However, for those with frontotemporal dementia with concomitant motor neuron disease, those people typically see a GP earlier, which is expected given that those symptoms consist of motor changes. But those with semantic dementia typically see a GP later. And when I say earlier and later, these are on average, say 10 to 12 months earlier and later on either side. And so this is of real importance when it comes to when can we intervene in terms of having better detection mechanisms in place so that this time to obtaining a GP referral is actually reduced or even detected prior to the effective onset of symptoms. And so much of the work that I'm doing here looks at this as a starting point in the pipeline, because a GP referral is really important for subsequent assessment that leads to a diagnosis. So it's one of the first stages. So in saying that, that's pretty much brings us full circle to all the questions that motivate me and some of the research I've done in this space as a function of these questions. Hope you found it interesting. Hope you learned a bit more about me. And if you have any questions, let me know. Thank you. Yeah. Nice. Thank you.
I'm interested in, you've obviously talked about body composition and dementia. Have you ever looked at, instead of looking at fat mass, have you ever looked at lean mass?
No, I haven't looked at lean mass. No, but there is some work being done in that space, but I haven't investigated it.
Just curious, I mean, if you look at fat mass, your station versus
yeah. Yeah, lean mass, like bone mineral density, and then interstitial fluid. So, but yeah, I mean, there's ways in which this could be investigated directly. Particularly, UK Biobank has data in relation to this. But yeah, it's definitely a space to explore. Yeah. Yeah, definitely. I think sarcopenia is established as another risk factor that we see, and I think in part it's falls risks that are associated there as well, and we see across menopause that women typically have lower bone mineral density. Like the typical recommendations from a GP is to engage in resistance training to help maintain that bone mineral density. But yeah, that decline in muscle mass occurs across the life course, so that would be interesting to pursue. Yeah.
Yes, Nick.
Thanks very much. It was very engaging and clear.
Thanks.
You said at the beginning of the talk that different people think differently about aging and dementia. So I thought I'd come back to that and ask you what you think, and whether what you think contributes more to the mechanisms that we end up using dementia.
Yeah, so hopefully sprinkles of what I think are in this talk in regards to the fact that it's the conversation around aging and dementia are confounded by lifespan, and in fact the processes that drive aging are what drive our risk for brain health. And so the degradation of these aging processes of clearance of waste and the ways in which we've fixed mutations that occur, that accumulation over time, it can be thought of as a wear and tear effect is what we see. And so that leads to the investigation of the processes that are closely tied to aging. And so the largest effects that we see in relation to dementia are age, sex, and ApoE status. And so within aging, I think the factors that are important to investigate are not only genetic factors, but also changes that co-occur with age. And that's where we talk about these sort of modifiable and non-modifiable factors that are difficult to tease apart statistically when age takes so much of the variance. And so if I was speaking to someone about focusing purely on modifiable factors, what are those that I think have the largest influence? I think a lot of the research shows rich social networks are really, really important for brain health. And that's shown indirectly through the fact that hearing loss is a risk factor for dementia. And so the people typically withdraw when they have poorer hearing, and so they're less likely to engage in that cognitive stimulation. In addition to what social networks provide for stimulation of the brain. In addition to that, I think that what the cardiometabolic factors, when you look at number 1 and number 2 as the reasons for death for men and women in Australia, that can't be ignored. And so, yeah, we've got to think carefully about, so what are the factors within these cardiometabolic differences that we see that contribute to the changes. And so much of my research now is really trying to identify which of these effects have the largest magnitude of effects, and with reference to the level of precision of those estimates, and considering things like sex differences that aren't accounted for in the Livingston paper. Like how do these effects differ by sex, given that it's such a big contributor to dementia risk? So those are probably the main things.
So given this, and given the slide you put on the screen showing that doesn't all happen at the end of life or even in midlife, and given type of factors that contribute to aging and development of dementia, behavior change is obviously a major opportunity to change these things, but how do you engage young populations in this space, given that they're generally not interested in what's going to happen in 50 or 70 or 80 years?
Yeah, you're right. I think creating a clear narrative that it's the accumulation of burden over time, and it's the small changes that accumulate to big differences across the lifespan. And so engaging young people in these behavioral changes, I think, is also important to balance these recommendations with something that's reasonable. I would never, there's there's a joke I think about very regularly, probably once a week when it comes to aging research, which is a man goes to his doctor and he asks the doctor, what can I do to live longer? And the doctor says, well, do you smoke cigarettes? And he says, no, I don't smoke anything. He says, oh, do you drink alcohol in excess? How many times a week do you drink? He says, no, I don't drink alcohol. Joke goes on into some other areas that I won't go for now for context, but at the end of it, the doctor says, so why do you want to live this long and boring life? And I think about that so often because it really touches on something true in humanity, which is that we want to live well. We want to delay these really nasty diseases or changes in brain health, but we also want to balance that. And so it really speaks to moderation. I would never say to someone, don't have crisps, don't eat junk food, but finding that moderation balance, which differs at individual levels, is, I think, a really, really important recommendation. So if there was someone who is drinking alcohol in excess, yeah, and it's also quantifying the effects of those changes. So for example, I love coffee. I drink several cups of coffee a day, and many times I've thought about its contribution to brain health and how it's a cognitive enhancer. And so I've gone through periods of my life where I've actually reduced my coffee intake to have its cognitive enhancing properties. So I drink once a week or once every few days and just use it when I need it. And I find, yeah, it boosts my cognition more noticeably, but I enjoy coffee. And so I lose that satisfaction. And so I'd say pick your vices carefully. I would rather have 2 to 3 cups of coffee a day and a non-alcoholic beer on occasion than 2 to 3 drinks of alcohol and decaf coffee, right? It's about being sensible with our recommendations. And I think sometimes people will tune out when we say recommendations such as don't drink alcohol or don't do this or don't do that. It's something where if you can make small changes to your current lifestyle that have a meaningful effect across the life course, I think that's valuable. That's my perspective, and people may differ. But